everyone. Welcome back to Vigyan Marg. In today's video, we are going to build a solid understanding of one of the most fundamental topics in microbiology, which is culture media. And if you have ever wondered how do we actually grow invisible microbes in the lab, and why do we use so many different types of media like culture, nutrient agar, blood agar, differential media, enriched media, and we will learn all of these things throughout this video. And this video is going to be very helpful for MBBS student, bachelor's, masters, and others entrance exam like NEET, GATE, and also for lab practical. This video is going to be very helpful, and we will start from the very basics, like what is media, what is culture, and what is agar, and we will go all the way to exam and viva level questions. So grab your notebooks, and because by the end of this video you will be able to understand to choose the right medium for the right organisms and for the right purpose. So let's begin our journey into the culture media in microbiology. So myself Arnav, and I am a student of IIT Guwahati, and I will be guiding you throughout this lecture. So let's begin. Before diving in, let's look at the learning objects for this session. By the end of this lecture, you should be able to explain why culturing microbes is fundamental to microbiology and how it underpins diagnosis, research, and industry. Distinguish defined media and complex media and understand when each is used. Compare liquid, semi-liquid, semi-solid, and solid media, and understand why agar is such a powerful solidifying agent. Classify media functions like supportive, enriched, and selective and differential media. Understand the enrichment culture. Classify media based on their function into supportive, enriched, and differential. And we will learn to how you will apply strict plate, spread plate, and pour plate techniques, and when each technique is appropriate in different scenario. And we will learn we will learn using standard colony morphology terms to describe isolated colonies in a way that is meaningful for identification for different microorganisms. So in nature, microorganisms almost never live alone. They are part of complex mixed communities in soil, water, uh, blood, and human respiratory tract, human gut, and so on. Studying them directly in such mixed environment is extremely difficult because many species are present in simultaneously and interact in a complicated way. So in the laboratory, culture medium allow us to grow organisms in a controlled and isolated condition. Okay, and where we can grow them in a pure form, manipulate the conditions, and observe the specific properties. Even though only about 1 to 5% of the microbial species are currently culturable, and on standard media, that small fraction includes nearly all medically important pathogens and microbes used in the industry. And application of the culture includes the antibiotic testing, pathogen identification, quality control, and different industrial fermentation process. Okay, so culturing microbe is not just a basic skill; it is one of the core enabling technology of microbiology. And next, let's fix some core definition that we'll use repeatedly throughout this lecture, like culture. What is culture? A culture is simply a population of microorganisms growing in or on a nutrient medium. Okay, and it can be pure culture, ideally derived from a single cell. Okay, and or a mixed culture containing several species in there. And what is medium? A medium or a culture medium is the nutrient material. It may be liquid, can be liquid or solid, and the that supplies the water, energy source, carbon, nitrogen, and minerals, and sometimes vitamins or growth factors required for the growth of the microorganisms. Okay, and next is broth. A broth is a liquid culture medium, and when organisms grow in broth, you see changes such as turbidity. Okay, when you grow microorganism in a broth, you will see turbidity in that broth and formation of a surface pellicle or sedimentation at the bottom. Okay, later in the lecture, terms like slant, plate will refer different physical presentation of some basic nutrient medium. And next, one way to classify culture media is by their chemical composition. First, we have natural or empirical media. Okay, here the exact chemical composition is not fully known. Okay, and they are prepared from natural materials like milk, blood, meat extract, vegetable juices. These media are often used for fastidious organisms and that require complex and sometimes unknown growth factors. And second is complex or undefined media. These are partially defined media type of, and they contain ingredients such as peptones, okay, meat extracts, and we know in general what they contains, like amino acids. They mainly contain amino acids, minerals, vitamins. Okay, and but not the exact amount of each component is not known properly. Example like nutrient agar, nutrient broth, MacConkey agar, and many routine diagnostic media. There is some batch to batch variation is also present there, but they are robust and expensive and support a wide range of organisms. And there is a third definition is defined or synthetic medium. Okay, in this media, every single component and its concentration are precisely known. Okay, and examples include Chapek-Dox medium, mineral glucose medium, and these are crucial in physiological and nutritional studies, where you want to ask, like example, what happens if I remove this amino acid, and can this organism use citrate or as its sole carbon source. So in defined media, the specific components are precisely defined. So natural and complex media are about richness and practically, while defined media are about to precision and experimental control. So in the next case, another simple but very important classification is by physical state, that is the consistency of the medium, like liquid media or broth, which generally contain 0% agar. Okay, and there is no solidifying agent present in there. These are used for bulk growth, for many biochemical tests, and very commonly for enrichment when you want increase the number of particular organisms. Okay, and another is semi-solid medium. Semi-solid medium generally contain 0.3 to 0.5% agar, and they are generally soft and almost jelly-like structure, jelly-like, because the bacteria can move throughout them, and semi-solid media are used for motility testing, certain transport media, and and occasionally for specialized enrichment, where a gradient of oxygen or nutrient is helpful. And another type is solid media, and solid media generally contain 1.5 to 2% agar. This form a stable or firm gel, and solid media are essential for isolating colonies. Okay, and plate counts, viable plate count, study colony morphology and pigmentation. Most of your strict plates, spread plates, and pour plates method are done on solid agar media. So the same nutrient formulation can be present as a broth, as a semi-solid, or as a solid plate simply by changing the agar concentration. Okay, and we move on to the next. Why is agar is almost universally used as the solidifying agent in the microbiology? This is the first question will come in our mind. So we will look a one by one properties of the agar. So first, look its thermal property. Okay, agar melts at about 95 degrees Celsius, which is high enough to allow sterilization in autoclave. Once melted and then cooled, it does not solidify until 42 degrees Celsius. Okay, and this is crucial because it gives a window where the medium, okay, where the medium is still liquid but cool enough to safely handle and to add heat sensitive components. Like if you want to add antibiotics, okay, and different components you can add in this temperature. Okay, and second is agar is not metabolized, okay, by the most bacteria. Is agar is not metabolized by most bacteria. The microorganism cannot metabolize the agar. That means it remains a chemically inert scaffold. Okay, and it remains stable throughout the incubation period. The bacteria grow on or in the grow on the agar, and they cannot digest it and liquefy the medium, unlike gelatin. And third is agar gels are stable at wide temperature range. Okay, and they stay solid at typical incubation temperature like 37 degree, 30 degree, and room temperature also. They remain intact in this temperature. And if we talk about the history, agar also introduced into bacteriology in the late 19th century by Fanny Hess. Okay, you need to remember this name, maybe come in the competitive exam. Who's who suggested that it who suggested that it as an alternative to gelatin in Robert Koch's lab. Okay, and gelatin melted at 28 to 30 degree Celsius. 28 to 30 degrees Celsius. This is the main drawback of gelatin. Okay, and also degraded by some bacteria, making it impractical for the experiments. Agar solved both problems and has remained the gold standard for the experimental analysis. And next, we look at some key ingredients that appear again and again in complex media and understand why they are used. So first is peptone. Okay, and peptone hydrolysate, partially digested protein from source like meat, casein, and they provide amino acids, okay, short peptides, nitrogen, and energy source, as well as vitamins. They are primary source of nitrogen in most complex media. Okay, nitrogen is the primary source. And next is yeast extract. Okay, extract, uh, extract of is aqueous extract of yeast cell. Okay, it is specifically rich in vitamin B, B1, B2, and niacin. as well as nucleotide and growth factors. Okay, and it is particularly important for supporting fastidious organisms and that cannot synthesize all their own vitamins. Okay, and next is beef extract. Okay, is a water soluble beef extract is a water soluble extract of beef tissues. It contribute minerals like calcium, magnesium, iron, and organic acids, amino acids. Okay, and additional amino acids also present there. Together with peptones, it enriches the medium nutritionally. Okay, and the last one is sodium thioglycolate. Sodium thioglycolate is a reducing agent, and it chemically removes dissolved oxygen from the medium. Okay, and lowering the redox potential and helping to create a anaerobic condition in the medium. You will see thioglycolate in media designed for anaerobes and demonstrating oxygen requirement in that condition. Create anaerobic helps to create the anaerobic condition in the medium. If you understand what each of these ingredient does, and you can look at the composition of an unfamiliar medium and predict what kind of organisms it will support, and then you can select the components according to your medium. Next is functional classification of the media. Most exam relevant and clinical relevant ways to classify media is by function, and in the functional classification the same medium could be simple in composition but used in a very strategic way. We will be talking about supportive or general purpose media, enriched media, selective media, differential media, and as well as enrichment broth. Okay, and the key idea on this classification of media is these categories are based on purpose, not on their chemical complexity or physical states. For example, blood agar is a both enriched and differential media. MacConkey agar is a selective and differential media. Understanding this overlap is important for your Viva question or exam related aspects. In the following slides, we will know each of these functional types of this media and specific examples, and we will know specific use of these medias. Let's begin with the simple or basal media. Okay, this is the most simplest form of media. This media contain the minimum ingredients required to support the growth of non-fastidious bacteria or organisms that do not have complex nutritional requirement. And examples include peptone water, it which contains only 1% peptone, and sodium chloride 0.5% and water. Okay, and another is nutrient broth. Nutrient broth is made up of the peptone water and meat extract of 1%. And nutrient agar, it is made up of nutrient broth and 2% agar. And semi-solid medium, it is prepared by reducing the concentration of the agar 0.2 to 0.5%. Okay, and the use of basal media. Basal media have several important use, like they are used for testing non-fastidiousness of the bacteria. They serve if if it grows well in the nutrient agar, it is like non-fastidious. Okay, and they serve as a base for the preparation of the many other media. Okay, and like by adding the blood, serum, selective agents or indicator, you can add into that. And another is nutrient broth is used for studying the bacterial growth or bacterial growth curve in the batch culture. Okay, and nutrient agar is a preferred medium for studying the colony morphology. Okay, performing the biochemical test such as oxidase, catalase, and agglutination test. and demonstrating the pigmentation or pigment production of the bacteria. Okay, and think of basal media like a plain rice. Okay, like like microbiologically, basal media like a plain rice, you can eat it alone. Okay, you can eat the plain rice alone, or you can make it complex by adding some other items in top of it. Okay, so you can think like that. Okay, so next is selective media. So selective media are designed so that only certain bacteria can grow, and the name is selective. Okay, like, this is selective for certain bacteria to grow on, and while others are inhibited by specific substances in the present in the medium. Okay, so if you see about some example like Lowenstein Jensen medium used for Microbacterium tuberculosis. Okay, and it contains malachite green and other component that inhibit contaminants while allowing slow growing Microbacterium tuberculosis. Okay, and another is thiosulfate citrate bile salt sucrose TCBS agar. Okay, which selects for Vibrio species, which is collected from stool sample. Okay, or environmental samples using high pH or bile salt. They can grow on this agar or media. Another is deoxycholate citrate agar or xylose lysine deoxycholate agar, both used for enteric pathogen like Salmonella, Shigella from the stool sample, and bile salts and dyes suppress the normal gut flora while allowing the pathogen to grow. Okay, and another is potassium tellurate agar PTA, used for Corynebacterium diphtheria, where the tellurate inhibit the most other bacteria and is reduced by reduced Corynebacterium diphtheria producing characteristics black colonies. Okay, you can see the black colonies on the agar plate. So the certain concept is that selective media contain one or more inhibitory agent like bile salts, dyes, high salt or antibiotics, and that suppress unwanted organisms enriching for the pathogen of interest. And next is differential media. Are These differential media are designed not so much to inhibit organisms but visually differentiate them based on their biochemical properties. Okay, so the main example is MacConkey agar in comes under differential media, which is both differential and low selective medium, work as a also low selective medium. It contains lactose. Okay, and it contains lactose and red indicator. Okay, red indicator and bile salts. Okay, organisms that ferment lactose produce acid which turns their colony pink to red. Okay, lactose fermentators produce pink color colonies in Escherichia coli, and it helps to identify non-lactose fermentators produce colorless colonies. Okay, like such as Shigella form a pale colorless colonies because they do not produce the acid need to change the indicator red to pink. Okay, red to pink. So we can differentiate between the lactose fermentator or non-lactose fermentator bacteria by using the MacConkey agar. So the general what is the general biochemical process then? Lactose fermentator produce the component which generally convert the red indicator to pink, so by which we can identify. Okay, and MacConkey also contain bile salt, like I already explained, dye, which weakly suppress many gram positive, making it low selective for gram negative enteric bacteria. Okay, already explained the compositions. And another is cysteine lactose electrolyte deficient agar, which also differentiate lactose fermentator and non-lactose fermentator. Okay, it is specially designed for urine culture. For the urine samples, it is specifically designed for. Okay, where it reduces swarming and allow better colony differentiation. So in differential media, the presence or absence of color change like homeolysis or specific reaction helps you to distinguish between groups of organisms on the same plate. And we can directly compare between selective media and differential media, which is a favorite exam question in many exam. In your university exam you can see this type of question in microbiology paper. So selective media is primarily aimed to control which organisms will grow, okay, while inhibiting other organisms. They contain inhibitory substances, okay, that suppress certain bacteria while allowing others to grow. Okay, and the main readout is the presence or absence of growth of the microorganisms. And some example is mannitol salt agar, okay, Lowenstein Jensen agar already explained previously. Okay, and this contain high salt selective for staphylococcus. And another is differential media, on the other hand, allow many microorganisms, helps to grow many microorganisms. Okay, and but include indicators that show viable reactions in that media, that reveal biochemical differences in between the organisms. The main function is how colonies look, the color change, homeolysis or colony appearances will be different. And some examples are MacConkey agar, EMB agar, blood agar. Okay, and some and the differential media, the pH indicator, blood cells and other substances act as a reporter mechanisms, and in that case selective media, high salt, bile salt and specific antibiotic dyes are act as a reporter or barrier. Okay, in some media like MacConkey agar, blood agar, both differential and at least mildly selective, which you should always mention in the exam when you will write. And next are supportive and enriched media. Okay, supportive media or general purpose media are designed to support the growth of many non-fastidious organisms without giving a strong advantage to any particular type. Okay, example like nutrient agar, tryptic soy agar. Okay, their primary use is to routine cultivation and maintenance of the culture, okay, in the laboratory strain and as a base for the specialized other media. And another type is enriched media, take a supportive base and add extra nutritional component, okay, like blood serum, uh, chocolate agar. Their purpose is to support fastidious organisms, those that will not grow or grow very poorly on simple media. We will look at examples such as blood agar, chocolate agar. uh, okay, and more detail we'll we'll learn more detail in the next slide. Okay, and supportive media generally we can mm, write here, like supporting medias are broad and help to general growth of the organisms. And in enriched media we have extra nutrients and to support fastidious organisms. Okay, and next we will see some examples of selective and differential media. Okay, like blood agar, which is a enriched and differential media. If we read the mechanisms, blood agar supports the growth of many fastidious bacteria. Okay, and these can be differentiated based on their ability to produce hemolysins protein that lies red blood cells. Okay, hemolysis appear as a clear zone beta hemolysis and greenish hollow around colony alpha hemolysis, like it's seen in streptococcus pyogenes and beta hemolysis. Okay, we will see in next slide of the pictures of this those things. Okay, and another is used in methyl blue, which is a selective media and also a differential media. You need to remember these things. Okay, is very important according to this medium. These two dyes used in Y and methylene blue inhibit the growth of gram-positive bacteria. They also react with the acidic product released by certain gram-negative bacteria when they use lactose or sucrose carbon energy in the source. I already previously explained how the red indicator convert to pink and how they will select it. And colonies of gram-negative bacteria that produce large amount of acidic products have a green or malachite scene, like fecal bacteria such as E. coli. And another is MacConkey agar, which is a selective or differential media. The selective components in MacConkey agar are bile salts and crystal violet, which inhibit the growth of gram-positive bacteria. The presence of lactose and neutral red, a pH indicator, allow the differentiation of the gram-negative bacteria based on the products released when they use lactose as a carbon or energy source. Okay, and another is mannitol salt agar, which is a selective and differential media. Okay, you can read it out. Another is we will focus specifically on enriched media. Okay, and first example is blood agar is prepared by adding 5 to 10% of sheep blood to the molten nutrient agar at 45 degrees centigrade. Okay, and it is one of the most widely used media in diagnostic bacteriology. Okay, and it is enriched because the blood supplies supplies extra protein, iron, uh, and growth factors into the media. So it is also differential because it allows you to observe hemolysis and alpha, beta and gamma hemolysis into the media. And another is chocolate agar is heated blood agar prepared by adding 5 to 8% of the sheep blood into the molten nutrient agar at 75 degree centigrade. Okay, so the red blood cells lies and releasing hemoglobin, some factors and factor X in the medium. Uh, the medium turns brown like a chocolate because these factors are freely available in chocolate agar and is even more nutritious than blood agar and can support highly fastidious organisms like Haemophilus influenza, which do not grow on plain blood agar. Okay, and another is Loeffler serum slope is contain serum is particularly useful for isolation of Corynebacterium diphtheria. Okay, and finally blood culture media, whether conventional bottles or automated systems, are essential for enriched broth, which nutrient and sometimes anticoagulants designed to support very low number of bacteria and blood samples. In enriched media, we add complex biological materials to support delicate fastidious pathogens. Okay, this is the main concept of enriched media. And another is enriched broth. Okay, enriched broth are liquid media, okay, added with some inhibitory agents which selectively allow certain organisms to grow and inhibit other. Okay, and this is important for isolation of the pathogens from clinical specimens which also contain normal flora like stool, sputum specimen. Example for enriched broth. Example for enriched broth are uh, tetrathionate broth. Okay, and tetrathionate and other components inhibit many competing coliforms and while allowing the Salmonella typhi to multiply. And another is gram-negative broth, which is formulated to favor gram-negative pathogens such as Shigella over the normal intestinal flora. And another is selective F broth, which also used for Salmonella and Shigella from the stool sample, and where selenite acts as a selenite act as a selenite act as a selective agent. And alkaline peptone water, okay, used for vibrio cholerae. Its high pH and composition inhibit the many other bacteria while help to grow the vibrio cholerae. Okay, the general workflow of the enrichment broth is the specimen, okay, and there is enrichment broth, and there is incubation period is there, and next is subculture. Okay, and into selective or differential media. Okay, finally you can isolate the colonies throughout this process. And next, these are the visually contrast of image of supportive and enriched media. You can see one side that you have the typical supportive medium such as triptych soya agar. It provides a rich but relatively simple environment that allow the many species to grow. TSA is widely used in both clinical and industrial microbiology as a general purpose medium. On the left side you can see the enriched medium is present there, okay, such as chocolate agar used to grow the fastidious organisms that will not grow on the TSA alone. Okay, the visual difference you can see in that picture. Okay, and next we'll move on to the next slide is uh, this is the summarize of the some selective media in a table format. Okay, for example, MacConkey agar. MacConkey agar is selective for gram negative, and because bile salts and crystal violet inhibit many gram positive bacteria, it typically used for stool and urine sample. And another is mannitol salt agar, which is selective for staphylococcus due to its very high salt concentration, like 7.5% sodium chloride, and other bacteria are inhibited like staphylococci, and which are salt tolerant can grow. And another is Baird Parker agar, which is selective for staphylococcus aureus, and it contains lithium chloride and tellurite, and as work as an inhibitor agent in that medium, is commonly used for food and pharmaceutical microbiology to detect. Sorry for the inconvenience. And which help to detect the food and pharmaceutical microbiology to detect the Staphylococcus aureus bacteria. And another is XLD agar, which is selective for Salmonella and Shigella. Deoxycholate and certain dyes inhibited normal coliform. Okay, and it is widely used for stool and food testing. Now, as you go through the exam, always link each selective medium to which bacteria it select for, and which agent make it selective, and which inhibitors are used. Like in these things you should think about, then you can recall these informations. And if we move on to the next slide, there is differential media, visible biochemical differences. Okay, so on blood agar, the key difference feature is hemolysis. Okay, hemolysis patterns. I will show you the pictures, then you will get better understanding. In beta hemolysis, a clear zone around the colonies you can see to compete the lysis of the red blood cells seen with organisms like staphylococcus pyogenes. You can see in the pictures beta hemolysis, you can see a clear zone of clearance, you can see. Okay, and there is alpha hemolysis, a greenish hollow form, partial lysis and oxidation of hemoglobin seen with streptococcus pneumoniae. Okay, and there is gamma hemolysis, there is no hemolysis occur in that case, and no change in the medium is seen. Okay, and on MacConkey agar, differentiation is based on the lactose fermentation. I already explained these things previously. uh, the lactose fermentation like E. coli produce a acid and form pink or red colonies, whether non-lactose fermentators like uh, Salmonella, Shigella form a pale or colorless colonies in the medium. And another is TSI agar, or sugar and iron sulfide, which is triple sugar ion. Okay, and differentiate enteric bacteria by their sugar fermentation pattern or hydrogen sulfide, and yellow slant or yellow indicate in yellow color indicates the sugar fermentation, uh, indicates the hydrogen sulfide production, which is typically the salmonella. Okay, differential media gives you an immediate visual clue to biochemical behavior even before you run detailed biochemical test. Okay, this is the advantage of this differential media. Next, if we move on to the next slide, different bacteria culturing aerobes versus anaerobes. Okay, different bacteria have different oxygen requirement, so culture condition must be adjusted according to that organisms. Okay, in the case of aerobes, aerobes which required oxygen to grow can be easily grown by sticking flask in the incubator, bubbling sterile or air through the large fermentator, or simply using normal aerobic plating on the surface agar. Uh, you can grow the aerobic microorganisms. Okay. In the case of anaerobes, however, either do not tolerate oxygen or are likely killed by this if the oxygen is present there. Okay, oxygen must be excluded or chemically removed from that media. Okay, and there are several reducing medias are used, like thioglycolate contain agents that remove the dissolved oxygen and create a gradient from oxygenated at the top of the anaerobic at the bottom. Okay, and there is another is anaerobic jars, which is gas packs, such as those using gas pack system create an oxygen free environment for plates by combining hydrogen with oxygen over a palladium catalyst to form water. Okay, and there is anaerobic chambers are also present there to provide a large and enclosed oxygen free workplace where multiple plates and tubes can be handled without exposed to the air. And another is anaerobic pouches allow individual plates to be incubated anaerobically using the chemical sachets that absorb oxygen. Okay, and additionally, the another broth is Robertson cooked meat broth. Okay, it contains chopped meat particle, beef or heart, which provide glutathione, a sulfhydryl group containing reducing substances, and unsaturated fatty acid. It is the most widely used anaerobic culture medium. Okay, the main concept from this is culture condition must match the organisms oxygen preference, and we have this method to perform those experiment. And another is an enrichment culture. Okay, strategy for isolation. And enrichment culture is a clever strategy for isolating a desired organisms from a mixed population by designing conditions under which that organisms has a competitive advantage. Okay, and for that we have to choose the inoculum source. We need to choose the appropriate inoculum source in that case. For example, if you want oil degrading bacteria, you take oil contaminated soil. Okay, and if you want phototrophs, you use pond water exposed to light. Okay, in that this is the examples, and you have to design the selective media. Okay, this is the main thing you have to do, that contains nutrient only your target can utilize and grow on that medium. For example, a medium where crude oil is the only carbon source for oil degraders, and or a defined mineral medium. Okay, which with a CO2 and light for phototrophs. Okay, you need to keep in these things in your mind. Okay, now you have to set up the environmental conditions the where the organisms can grow freely. Okay, such as you have to maintain the pH, the temperature, the oxygen level. Okay, and the light that closely mimic the natural niche of the your target organ, like the natural conditions where the microorganism generally used to grow. You have to maintain that particular conditions in your uh, laboratory. Okay, after 1 to 4 weeks of incubation, the desired organisms becomes a major component of the culture. Okay, you still need to subculture onto the plates, different plates. Okay, and perform pure culture techniques like strict plating. But now you are starting from a much more favorable mixture. Okay, for that case, enrichment culture is therefore a pre-isolation step. Okay, before you are going to pure culture, this is the pre-isolation steps. Okay, especially important for environmental or ecological microbiology. Now pure culture techniques. Okay, to study a microorganisms in detail, we need to do pure culture, a population derived from a single cell or a single colony free from contamination, like a single colony of particular microorganism is present in that particular medium. Okay, so there are different methods like strict plates, spread plates, pour plates techniques are available there. All three methods share a common goal to separate the individual cells sufficiently on a solid surface so that each one can grow into discrete or well isolated colony. In the next slides we will look at each of these techniques, their step by step procedure and their advantage and limitations. So let's see, strict plate technique. Okay, the strict plate technique is the most common used method for obtaining the isolated colonies of from a particular mixed culture. Okay, the principle is very simple. By sticking a loop full of culture. Okay, a loop full of culture and flaming the loop between different time period, and you progressively dilute the number of cells being deposited until individual cells are left well separated. Okay, first what will you do in the stick plate? Okay, first you have the loop. Okay, you need to flame the loop. Okay, next you need give some time period to, now you have to write properly. Now you have the loop, you need to flame it, then you should give some time to cool it down, and you have a broth, the tube. Now you will pick up the mixed the culture, and you will strict it in the your plate. Okay, now for the streaking case you can divide in two coordinates. Okay, you can divide in two coordinates, like this, or you can divide into four coordinates. Okay, I'm showing only for the two coordinate. Like when you take the culture, you will start from this point, you will strict it like this in that medium, and again you can, where you have end it, and again you can flame the loop. Okay, and again where you have ended it, and from that you can start it like this. Okay, this thus you can repeat, you can make, if you make it a four quadrant, you can repeat in the same, in that case same thing you can do. This is the main advantage for this. This is very simple, very fast, and uh, require no special equipment. It is ideal for a routine diagnostic in lab. The limitation is are there, uh, that is qualitative and not quantitative. Okay, and you cannot calculate the CFU. Okay, I will explain it later, the CFU, coloniformic unit, and CFU cannot calculate, and the results strongly depend on good techniques, especially proper framing and minimal overlapping of the streak, how you are streaking. These things are depend in that case. Okay, this is the visualization how you will streak. You will take the loop, I have shown you before. You will flame it, and you will streak it, strict it accordingly to your preference. Okay, this is the result you can see in the plate. You will get these types of colonies in that stick plate technique. And next is spread plate technique. Is the best when you already have or can prepare a known dilution of your sample, and you want to quantitative the result in terms of your coloniformic unit per ml. Okay, the principle is to distribute a small known volume, usually 100 microliter, okay, of a diluted sample evenly over the surface on a agar plate, and then count the colonies that rises on the plate. Okay, for that case, how will you prepare the sample? For that case, prepare the dilution. First you have to prepare the dilution. You have to do the serial dilution. Like how will you do? You have a one tube. Okay, you have the culture here, main stock. You have the main stock. Okay, this is the main stock. Okay, next in that second tube, what will you do? Suppose you have 9 ml of H2O in there. Now from that you will take 1 ml of the culture, you will add it here. Okay, now this is becomes your 10 1/4 dilation. Like the same thing you will do it from one, you will take from 1 ml from the this one, and this will become your 2-fold dilation, so on you will do it the dilution, and it depends on how many folds you are diluting the sample. Okay, like this. And you have to now sterilize the spreader. Now you have a spreader, in that you have the spreader for the spread plate technique. You need to dip into the ethanol and flame briefly and give some time to cool it down. Okay, now spread the spread evenly throughout the plate. Okay, you have to spread throughout the plate evenly and incubate for 24 to 48 hours. Now coming to the calculation part, colony forming unit. Okay, now what is the formula of colony forming unit? CFU per ml, where you will write number of colonies into dilution factor divided by volume. The volume is plated in ml. Okay, suppose you got in your spread plate, you got 2 3 4 5 colonies. Okay, now you have 5 colonies and your dilution factor suppose 6. Okay, and you plated 0.1, okay, or 0.01, 0.01 ml. Okay, now you can calculate your CFU from that. Okay, this is the formula you need to remember. This is very important. Okay, next, this is the split plate method. Two types you can do it. A pour plate method is also present is there. A split plate method is also there. In the case of pour plate, you inoculate the your microorganism into the broth, and then you pour it into the plate. Okay, and then you swallow or mix it evenly, and now you left it for incubation to grow on the microorganism. And the spread plate, I already explained to you how we will spread it, and this is the colonies you can found in the spread plate technique. And pour plate method, the pour plate technique is another quantity method, but here the bacteria are mixed with agar rather than being spreaded on top. The principle is you transfer 1 ml of diluted sample into the empty sterile petri dish. Okay, you can do it like that. You can transfer the 1 ml of diluted sample into the petri dish and add the your agar into that and mixed it accordingly. Others, what you can do, you can first mix it into the your agar, and you can pour it into the plate. Okay, both things you can do it. As the bacteria grow, colonies develop both on the surface and within the depth of the agar. The surface colonies appears smaller, lens shaped, or embedded. Okay, the advantages include like better for some organisms that spread excessively on the surface, such as swarming proteus, because being in the agar restrict the movement of that bacteria. Okay, that's why you should spread it on the top. And it can be helpful for heterogeneous samples, and certain specific regulatory methods prefer pour plates. The disadvantages are that some bacteria may be heat sensitive and damaged by the 45-degree agar temperature. When you are mixing it, the temperature of agar is 45-degree. Okay, in that case the some bacteria can be heat sensitive, they can get damaged. Okay, and subsurface colonies are more difficult to pick for subculturing. It's difficult to pick from that from the pour plate technique. Okay, you can see the pour plating. I already explained to you how you will do the dilution. So on, you can do it like up to tenfold dilution. You can do it tenfold according to your experiment, or according to your requirement. You can do it. Okay, further on. And this is the plating method. You will now mix the culture with the broth and will plate it into the agar. You can see different types of colony after the incubation period. You can see. Now if we come to the next case, like colony morphology. Once you have isolated the colonies, you can describe their colony morphology using a standard identification or visualization method, you can say. This is often the first clue in identifying microorganisms. Like in the case of form, the form can be circular, irregular, rhizoid, or filamentous. You can see in this picture. like circular, filamentous, irregular, rhizoid, spindle form. You can identify the microorganisms by visualize the formation of the organisms. Okay, the morphology actually. And the margin can be entire, undulate, erose, and filamentous. Like entire means smooth, undulate means wavy, wavy, and erose means serrated, and filamentous. And on the case of elevation, you can see the flat, convex, pulvinate, and ambonate. You can see like that. You can see the elevations: flat, raised, convex. You can see the morphology. And there is surface should be smooth, rough, wrinkled, or mucoid. Opacity: opaque, translucent, or transparent. You can see the color: white, yellow, orange, purple. You can see, identify by the color. You can see here some morphological identification is presented here, the different morphology present in different microorganisms. Okay, so in the next case we'll see why does growth vary within the colony. Okay, and even within a single colony conditions are not uniform. Okay, and this leads to a variation in growth and physiology across the colony, like in a nutrient gradient. Okay, at the edge of the colony, fresh nutrients are readily available. Toward the center, nutrients have been depleted. In the case of nutrient gradient, you have to mention the factor nutrient gradient. Okay, and another is oxygen gradient. Oxygen gradient in thicker colonies, the surface and the edges are more oxygenated. Okay, while deeper layers are, the core may become relatively anaerobic or less oxygenic. Okay, you understood? This is oxygen gradient. You have to think about it. And the waste accumulation, as the in the case of in the incubation period, the waste accumulation happen, and in that case the metabolic waste products accumulate more in the center. Okay, and creating a more hostile microenvironment. Okay, and the edges will be clean and center will be toxic metabolism will be accumulated. Okay, and the edges in the nutrient you can see the edge, in the case in the edges micro in the plate, in the edges the fresh nutrient will be there, and in the center the nutrient will be, consumption of the nutrient will be depleted. Okay, and in the cell age differences, and the cell age differences, cell at the age are younger. In the, if you see, in the orny, the cells present on the edge, okay, they will be more younger and often grow exponentially. Okay, while cells in the center are older, frequently in stationary phase or death phase. Because of these gradients, okay, because of these gradients, because of these gradients, cells in different regions of a colony may express different genes, have different sensitivity to antibiotics, and show different metabolic activities. This is one reason why microbiology protocols often specify using fresh media and young colonies. Okay, and for biochemical test and susceptibility testing. And now real world application. Everything we have discussed about media and culturing comes together in the application part, especially in the pharmaceutical microbiology. Okay, the key components is microbial limit testing. Okay, this is a regular standard like USP 61 and European Pharmacopoeia 2.6.112 describe the microbial limit test. Okay, for non-sterile pharmaceutical product. This is the total aerobic microbial count TAMC, which uses a general purpose medium like tryptic soya agar, okay, TSA, incubate 30 to 35 degrees centigrade for 3 to 5 days. Okay, and with this limit such as less than or equal to 10 to the 4 CFU per gram of product, depending you can get depending on the formulation. Okay, and next is test of test for specific microorganisms, TSM. Okay, in that case which check for specific pathogens such as Escherichia coli, Salmonella, তারপর Staphylococcus aureus, and Pseudomonas aeruginosa. Here the acceptance is typically absence of the colonies is there. There is no colony, you will not get 0 colonies as per the quantity you plated. Okay, in this context the choice of the medium, incubation condition, and even enrichment steps are tightly defined. Okay, and demonstrating how core microbiology principles are embedded in regular practice, you can understand by this experiment. And next, real life laboratory workflow for detecting salmonella contamination in a pharmaceutical products. Like if we use this as a example, previously what we have learned, we will see how the things is done. In that case the pre-enrichment culture is done. Okay, pre-enrichment is the buffered medium is used, buffered peptone water. Okay, this helps recover the stressed or injured salmonella cells without strong selective pressure. Okay, you recover the injured salmonella cells. Now next stage is enrichment stage. In enrichment stage, uh, tetrathionate or selenite broth, selenite A broth, okay, mainly selenite A broth is used. These broths contain inhibitors that suppress the competing coliforms and while allowing the salmonella to grow or proliferate, you can say. And another is isolation step. In the case of isolation step, XLD agar or brilliant green agar is used, and on XLD agar, salmonella typically appears as a red colonies, okay, with black center due to hydrogen sulfide production. On brilliant green agar, salmonella produce characteristics pink white colonies surrounded by red medium. Okay, already explained both this condition of the medium. And there is confirmation test. Using the TSI or MBBS or serological test, you can confirm the identity of salmonella. Okay, this example ties together everything we have just discussed: selective media, differential media, enrichment culture, pre-enrichment, and the logic of the microbial testing in the real laboratory context. Whatever we previously learned, we already how we will apply those things I explained in this slide. I hope you understand. And next, some quick exam tips. Okay, and common questions you can see here. Why peptones used in the media? Okay, you can give me the answer in the comment box. Okay, and why is agar used as a solidifying agent? Difference between selective and differential media. MacConkey agar is a dash and dash. Okay, uh, which plate method is best for CFU count? Okay, and what is pure culture? Definition of pure culture. Comment down these answers. So to summarize the entire session, the key things you need to remember, like culture media is the foundation of microbiology. Okay, choosing or designing the right medium is essential for the isolating and identifying and studying the microorganisms. Okay, you should be comfortable with the big four functional types. What are those? Supportive, enriched, selective, and differential media. You need to study these things very briefly. Okay, Master the three isolation technique. You need to learn strict plate, spread plate, and pour plate. Okay, strict plate is fast, spread plate is quantitative, and pour plate is protective, also quantitative. And understand the agar properties. Okay, especially its melting and solidifying temperature. Okay, and inertness of the agar, like how when you increase the agar concentration. Okay, as you will if you increase the agar concentration like 1.5 to 2, how the inertness will increase. Okay, you need to study all these things. And recognize the colony morphology. Okay, provide the crucial early steps for the identity purpose, and when combining with the appropriate biochemical test leads to a accurate identification of the particular microorganisms. If you can explain all these points with example, you will not only handle exam questions confidently but also be able to apply this principle in your laboratory work. Okay, so I will sum up here, and thank you for joining this session, culture, media, and microbiology. If you found this helpful, please like and don't forget to subscribe this channel. So further I will make future more videos like on the topic of how we'll identify the identification of bacteria, like AST method, molecular diagnostic test, and all of which build a solid understanding of how we grow microbe in the lab, and how we will identify those microorganisms in the laboratory by different using by different biochemical testings. Okay, and thanks again for watching, and happy studying, and don't forget to subscribe this channel, and you can also join the Telegram channel. I will pin down the link in the description, and please subscribe. And if you like this video is this if this video is helpful for you, please don't forget to like. Okay, thanks for watching.